On this week's OIS Retina podcast, we have the privilege of talking to Steve Pakola, MD. Steve is an industry veteran who has held key leadership roles in preclinical and clinical development, regulatory affairs, and medical affairs. Currently, he serves as Chief Medical Officer at REGENXBIO, a leading clinical stage biotechnology company seeking to improve lives through the curative potential of gene therapy.
Hello, welcome back, everyone. This is Firas Rahal for the OIS Retina podcast. I'm a partner at Retina Vitreous Associates here in Los Angeles, where I'm based, and I'm a partner at Excite Ventures, which is based in New York, but has multiple locations. I'm very happy to have as my guest today, Steve Pakola, who is a doctor, an MD, but he's in the business side of things, at least at this point in his career. He's the Chief Medical Officer for REGENXBIO. He's had a number of other positions, which I'll go over in a minute. Hello, Steve. Welcome. Thank you for coming on.
Hi, Firas. Thanks for having me.
So, Steve, I went over some of your history, and I know a little bit of it from before because we've met and talked before and in this context and others. It looks like you have a very long history in biopharma and drug development. I saw that you were in the cardiovascular space with Boehringer Ingelheim, which, as I recall, I grew up in Danbury, Connecticut. They had a big office building in Danbury. I don't know if you were.
That's right. Situated. Yeah, the Ridgefield Danbury office. That's where exactly. I was at. Correct.
Is that where you were?
Yeah, that phase. So most of my drug development experience has been ophthalmology and mostly retina, but I did do some cardiovascular work, which was fascinating. And it actually, interestingly enough, was my experience in cardiovascular that led to me going to Thrombgenics, which was originally a cardiovascular focused company based on their work with microplasmin.
I didn't know that. I mean, I know the product Jetria, and I know the microplasmin story, and I've used it myself a number of times. We were in the clinical trials in our group. Back then. I know you are with Ram Pawanki now, and he was part of that group as well. I didn't know it started in cardiovascular space. What was it intended to do there?
Yeah, it's fascinating. The founder, Desiree Collin, a physician scientist from Belgium, who had a lot of the initial IP around TPA, tissue plasminogen activator. So his mission was to come up with newer and better thrombolytic agents. Okay. So that's really what Thrombgenics started out as. And when I joined the company, they were advancing microplasmin as a thrombolytic. When I joined, I was aware of work that Mike Tracy and George Williams had done with autologous plasmin.
Sure.
And given my original training in ophthalmology, I kind of put two and two together thanks to the work that those guys and others had done. And long story short, over a matter of a few years, we morphed into an ophthalmology company because we realized the potential to advance and really make a difference with microplasmin actually made more sense in terms of treating vitreoretinal traction and macular holes. So that sort of got me back away from cardiovascular back into ophthalmology.
And you ended up
from there. Ram and I,
a 10-year run, and then you were with Airpio as well.
Yeah. So then, after Jetrea advanced, it got approval. Of course, I then moved on to a couple smaller ventures. So really seeking to see if I could do it again or be a part of earlier stage programs and advance them into and along the clinic. So Amichem was a company focused on glaucoma treatments, and particularly the Rho kinase pathway. We ran into roadblocks there like others have, where yes, indeed, we showed in the clinic we could lower IOP, but not quite enough to really be an advance for treatment of glaucoma. And then I moved on to AirPO, where we were working on Tie2 activation as a treatment for retinal vasculopathies like diabetic retinopathy.
Yeah.
And that was until a year and a half ago when I joined REGENXBIO. And you mentioned Ram Polanki, who'd already joined REGENXBIO about 6 months before I had. And it was from speaking to Ram and others that I realized the excitement around the platform that exists here in terms of gene therapy, not only for treating monogenic disorders, which is kind of the traditional approach of using gene therapy, but also an innovative approach of actually using gene therapy to deliver a therapeutic protein to treat more common diseases like wet AMD and diabetic retinopathy. And so I joined on about a year and a half ago, and it's been a great ride so far, where we've been advancing our lead program, RGX-314, for treatment of wet AMD, and more recently, treatment of diabetic retinopathy. And what really kind of excited me is kind of a unique aspect of people hear gene therapy and they think, oh, that's risky, or, how do you know it'll work? And yes, any kind of new modality, the probability of success you have to take into account. But on the other hand, of the potential targets that you could go after, anti-VEGF and targeting VEGF with gene therapy, what a great validated target that we have based on everything we know about wet AMD and diabetic retinopathy and the other VEGF-driven retinopathies. So really a neat opportunity to advance gene therapy in a place where you expect anti-VEGF to work based on everything that we know. But the big unmet need now we're seeing is the unsustainability of the frequent injections that you and your colleagues have to go through indefinitely, and your patients, and that in the real world, the patients aren't getting the injections they need because of the treatment burden that exists with this. So certainly I've seen that in my years, the other companies going after more durable treatment options, like some of the agents that are in development now. So really just excited to work on something that is kind of the ultimate in durability, which would be a one-time treatment that you could really address the treatment burden for these patients.
Well, gene therapy is such a broad thing, and I'm definitely going to get into some specifics with you. We have you, and we have your expertise. I want to pick at that a little bit. It's definitely sexy and exciting, and for all the reasons you said in our space, the durability factor. But it also sounds like a unique way to really treat disease. And a little later, I want to get into this monogenic versus like a wet AMD concept. But since I'm sort of novice, and I think a lot of people who are listening won't clearly understand it at your level, but maybe you can help us understand some of the basics. Let's start with the basics. This is an AAV vector that your company is using. There's some newer ones I read about in your program. These are non-replicating viral delivery vehicles. How does this work sort of in a broad sense so we can understand? Is there a capsid? Is there DNA material? How does it then get in the cell and do what it's supposed to do? And then ultimately, why are these current versions better than earlier versions?
Yeah, it's a key question. So you say gene therapy, but ultimately you got to find safe and effective ways of doing that. And a lot of what we've learned in the field over the years is that it really comes down to the components of how you deliver it and to see how effectively and how safely you can do it. So to start with, gene therapy you can think of it first as either ex vivo or in vivo gene therapy. So you'll have heard, and the listeners will have heard about CAR T therapy and some other therapies for blood-borne lymphomas, leukemias. Those are ex vivo gene therapies where you actually take the patient's cells and you modify the genome ex vivo and then put their cells back into the patient. And there have been a few approved therapies like CAR T for this. What we're doing is true gene therapy where you actually give a vector with the gene to the patient to actually deliver the gene to the patient's own cells so that those cells can actually produce the protein based on that genetic material, coding material. And within the in vivo gene therapy space, you can think of two main different settings of what you're going after. One is generally rare monogenic disorders where a patient's own particular single gene is either missing or non-functioning, and that leads to a monogenic disorder. And the goal there is to use gene therapy to actually deliver a functional copy of that gene into the patient's cells and into the nucleus of those patient's cells to produce that missing or malfunctioning protein. And that, of course, a famous example in our space, of course, is Luxturna for treatment of biallelic RPE65 LCA, which was really a milestone for not only retina gene therapy, but overall gene therapy around the world as the first in vivo gene therapy approval. Now that's kind of the traditional approach or the way people commonly think of gene therapy, but then it does open the door for, hey, maybe gene therapy can be used to address more common complex disorders that are multi-genetic, things like wet AMD and other VEGF-driven retinopathies, where instead of delivering a missing gene, you deliver a therapeutic protein. So in our case, we deliver the transgene is a monoclonal antibody fragment that is extremely similar, only one amino acid different from ranibizumab in our particular case. So one aspect is having the right transgene to produce the protein that you want. And then the other issue more broadly, or one of the other big factors is, well, what vector do you use to actually deliver that? And over time, the most common and the most tested and the most validated approach has been using viral vectors. And the reason this has worked out well is viruses, to survive, have developed a pretty neat mechanism of replicating in a host cell system, where basically, if we go back to our immunology, virology lecture hall, you have a virus such as the AAVs that are basically an outer shell that's the capsid that's made up of the capsid proteins. And basically that protects and it houses the genetic material of the virus. And what's interesting is that genetic material inside encodes for the proteins that make up the capsid shell, as well as the proteins that are required to allow the virus to replicate. So it's an ideal nature-derived approach that you basically hijack the viral machinery to allow to use the virus as a vector to deliver to the host cells, and then the virus is enveloped in an endosome and actually can get into the nucleus, where fortunately in the case of AAV, it doesn't integrate into the host cell's DNA. It just exists as an episome of genetic material. And of all these different viruses, you basically want to find one that isn't pathogenic, and ideally one that doesn't integrate into the host cell DNA, because you don't want to have the risk of it creating other problems by getting into the host cell DNA itself. So those are a couple of the factors that make AAV preferable to other vectors that people have tried. One, we know AAVs are non-pathogenic. In other words, they don't cause disease. So that's a positive. We know that they're less immunogenic than other vectors like adenovirus. So these are some of the factors that first led people to advancing adeno-associated virus vectors. And you mentioned there's a lot of numbers that go along with these based on how many of these have been discovered. So the first generation of AAV vectors are AAV1 through AAV6. And the one that most people are familiar with, because it's the one where most of the work has been done, is AAV2. And that actually was the vector that was used for Luxturna, for example, as well as a lot of the other initial clinical trials that were done for retinal diseases. Now, I did mention that these are less immunogenic than, say, adenovirus, but it turns out that we've learned a lot since the initial discovery of those initial viral vectors, and that's actually the origin of our company, REGENXBIO. So if I give you a little quick history, in the early 2000s at the University of Pennsylvania, Jim Wilson, one of the pioneers in gene therapy, discovered over 100 additional AAV vectors that have various additional numbers beyond those original ones. So these are all referred to as different serotypes. And the reason these are important is each different serotype has different characteristics based on their capsids. So that becomes very relevant because those different characteristics lead to different characteristics in terms of how well do they lead to transduction of different cells? How well do they lead to protein expression? How durable is the protein expression over time with different viral vectors? Even though generally AAVs are less immunogenic, we've found that a lot of these newer generation vectors, our platform technology, we call these the NAV platform technologies of next generation vectors, that these have a lot of favorable characteristics. And the ones that we've focused the most on based on what we've seen and what we've characterized are AAV8 and AAV9. So AAV8, for example, is the vector that we use with our lead program, RGX-314. And AAV9, which is the vector that we use in our non-ophthalmic CNS platform gene delivery program. And the reason we've honed in on these two particular serotypes is these positive characteristics. So we've seen with both these vectors much higher protein expression and much more durable protein expression than was possible with the first generation vectors. And we've seen that they have a lower immunogenic potential. So these are all factors that allowed us to advance not only our ophthalmic programs, but also our non-ophthalmic programs.
And the determination about immunogenicity and protein expression, in light of what you just said, is this just lab experimental trial and error? Are there clues to which ones may be better or worse in advance, or do you just have to put them in the lab and test them individually?
Yeah, it's a great question. I think putting it in the lab is the best way because you really see in a in vivo setting. There's been a ton of work over recent years in different ways of trying to improve these different characteristics of the vectors. Other approaches that people have tried have been accelerated evolution approaches, where you just try to create a lot of different mutations over time in an accelerated fashion. But ultimately, then you have to test, well, do those changes make
did it work?
Particular vector less immunogenic or allow for greater expression? The other key factor I forgot to mention is also tropism. It's not just a matter of how well you can transduce cells, but for different diseases, you want to have greater selectivity to be able to target specific tissues. And AAV8 and AAV9 are good examples of that, where we know we get very good tropism towards retina, for example, as a target tissue. And for AAV9, very good target selectivity or tropism for neural tissue, including the CNS. So to answer your question, you really got to test it. And we've seen also the hard way over the last couple of decades that you can see very encouraging signs in small animal models, and then you go to a non-human primate and those apparent benefits disappear. So it's been a long slog.
In all the pharma products too, same thing, right? You sometimes get blindsided by something you didn't expect, and I guess it's the same here.
Yeah, and that's why for us, we've evaluated all these in small animal models, but also large animal models, including non-human primate, to really confirm these favorable characteristics. But then ultimately, you got to translate into the clinic. And we're starting to do that in both our retina platform as well as our non-ophthalmic CNS platform, where we're demonstrating clear proof of concept in terms of not only being able to deliver the protein, the gene that then allows the cells to produce the protein, but we're seeing good durability after one-time treatment, and we're seeing that translate into the pharmacodynamic outcomes that we want to achieve by delivering those proteins.
That's very clear and great, and I want to get into the clinical trials and your clinical programs in just a second. I had one other question. You said something that was educational to me, and again, I don't have a good background in this. The viral vector can commandeer the transcription making processes of the cell without incorporating into the host DNA. Are you saying it gets into the nucleus and just commandeers the mechanisms without doing that, or can it happen both ways?
Depending on the vector, it can happen both ways. So, for example, lentiviral gene delivery does actually integrate into the host cell DNA. But that then creates its own known of where is it integrating, what might it be knocking out or interfering with, or what unknown outcome might that lead to. One benefit with lentiviral vectors is they have a larger capacity that you can insert much larger genes. So some programs have gone to lentivirus if they have very large proteins, and also for the ex vivo gene therapy approach that I mentioned, you see that used. But for in vivo gene therapy, you'd prefer a vector like AAV that doesn't integrate into the host DNA. So AAV is single-stranded DNA genome, and then you put your transgene in there and you take out the replication genes so that you have the non-replicating genome, and the virus is efficiently delivered into the nucleus. And just as you said, it just commandeers the basic transcription machinery to allow production of the RNA, et cetera, to allow for the eventual protein to be made in the cytoplasm. But it exists there without integration into the host DNA, correct?
It's just brilliant, really. I mean, this is just brilliant stuff, and I'm glad you explained it to us, to me and to the listeners in that way. Let's talk a little bit about the clinical programs, because I know you want to tell me and I want to hear about it, and this is what probably most of our listeners are hearing about. RGX-314, it's the one for a couple of different indications, wet AMD and diabetic retinopathy. You mentioned it, but which proteins are being made here? This is a ranibizumab-like protein that's being made, and you're sort of creating a biofactory. What's the target cell?
Yeah, so that's right. It's a Fab, so an antibody fragment, and the amino acid sequence is virtually identical to ranibizumab. It's literally only different by one amino acid, which is based on the reality of, if you think of a Fab molecule, you have the light and heavy component of that, and we produce both of those components as one protein that then gets cleaved to allow it to self-assemble as the antibody fragment that then has the ability to bind VEGF. And it's because of how that cleavage occurs that you have a difference of a single amino acid, but we see the same intended effect in vitro and in vivo. So where are we targeting? As I mentioned with our lead program, we want to target the retina. So we deliver subretinally, and that's what we've done to date. For example, in our initial first-in-human study in 42 patients that included doses across 5 different cohorts, we administered subretinally, and what we've seen preclinically is with that route of administration, we have very good transduction of the RPE and the photoreceptors, as well as some of the other layers, but it's really predominantly RPE and photoreceptor. And just as you mentioned, the idea in this case is to allow the patient's own retina to serve as a biofactory to produce this therapeutic protein at a steady level, and therefore obviate the need for repeated peak and trough levels with injection of the available anti-VEGF treatments. We've seen very good proof of concept from this initial 42 patient trial, where in our dose levels 3, 4, and 5, we've seen a dramatic reduction in the need for repeat injections, with many of the patients needing absolutely no further anti-VEGF injections. And importantly, this reduction in treatment burden has been achieved while maintaining or improving visual acuity, as well as anatomy as measured by central retinal thickness and assessing for actual fluid. So based on those results, we're planning to proceed into pivotal clinical development starting at the end of the year. We'll be announcing the details of that design later this year. In parallel with advancing subretinal delivery, we're also kicking off clinical development of suprachoroidal delivery of RGX-314. So the same exact molecule, RGX-314, but delivered into a different route or different plane via the SCS microinjector from Clearside, which we're in collaboration with for advancing this. And there as well, what we've seen preclinically is good transduction of the RPE and the photoreceptor. So the same target tissue and retinal layers, but just from a slightly different plane, but still fortunately very close to that target tissue. And the thing we like about suprachoroidal delivery is, one, it is close to your target tissue, so you don't have to give massive amounts like you do with intravitreal administration, because with intravitreal administration, you have to give orders of magnitude more, so that just to allow enough to diffuse to the back of the eye and to also get beyond the internal limiting membrane barrier to get enough transduction in the retina. And invariably with those high doses with intravitreal administration, you wind up with inflammation. So for those two reasons, we chose to go with suprachoroidal delivery for a office-based treatment option to deliver RGX-314. And we've seen preclinically that we do not have inflammation with suprachoroidal delivery, including in non-human primates. And it's based on that work that we then advanced to IND-enabling studies. And we actually just earlier this month announced the dosing of the first patient in our AVIATE study, which is evaluating suprachoroidal delivery of RGX-314 for treatment of wet AMD. And that's actually the first patient ever dosed via this route of administration with gene therapy. So a lot going on in both facets. We basically look at the subretinal delivery route as really the bird in hand, given the fact that subretinal delivery really is the gold standard in terms of safely and efficiently delivering gene therapy to the retina as a target tissue. And especially now that we've even shown this with our RGX-314 program with subretinal delivery for wet AMD patients.
I agree about your comment regarding the gold standard. At this point, a lot of things could turn out to be the gold standard. And as a vitreoretinal surgeon, I like the subretinal delivery. It doesn't seem like a complex procedure compared to other surgical approaches to gene therapy, implants, and so forth. And I'm involved in some of those trials as well. Which brings me to the question, is the move towards suprachoroidal, at least to study it, let's not say you're moving in that direction, but you're planning to study it, is that more, and I don't mean this negatively, is that more marketing so it can be said to be an office procedure, or did your surgeons or your trial results lead you to believe that there are safety questions about vitrectomy with subretinal injection or instrumentation might not be the best answer, or is it more of a marketplace question?
I'd say on the clinical experience that we have to date, we've been very encouraged. Our retinal surgeons in our phase 1/2 study have not had an issue with delivering the drug. So they've had an experience very akin to how you described it, where relative to what you all do as surgeons, this isn't a complicated procedure. And certainly you all have experience of creating or injecting subretinally, for example, with TPA,
Sure.
treat subretinal hemorrhage. It's a little more involved than that, in this case, there isn't already an existing a hemorrhage or
Right, you have to create the bleb completely.
So the surgeon does have to create a retinotomy and go in.
Yes.
But we've been very pleased with how the surgeons are able to do this. I think one interesting aspect is it's also much easier than historically has been the case for treatment of inherited retinal diseases. So there, for example, the Luxturna case, there are certain centers who do those, but there's a reason for that, in that with certain inherited retinal diseases, since you're replacing a missing gene or a non-functioning gene, there's an actual need to get the vector to where the action is, to the macula and the fovea, because you really need to get the protein produced in that very local area in each of those individual cells within the retina that you're targeting. The other aspect, of course, is a lot of these are infants and young kids, where the vitrectomy before you do the injection is inherently much tougher in young kids. And also that retina may be more friable right in that macular region in some of these inherited retinal diseases. One of the things we learned in doing this trial and working with our investigators is that because we're providing a therapeutic protein, and the patient's retina is going to serve as that biofactory to produce anti-VEGF that will serve as a reservoir basically in the vitreous, in the retina, we can produce that subretinal bleb anywhere. It doesn't have to be close to the macula. So for all those factors, it makes it a less risky procedure and a more straightforward procedure we've seen. And in our 42 patients, there was only a single patient who didn't get a complete dosing, and that happened to be a patient in one of the earlier cohorts, where the surgeon created one retinotomy, wasn't able to actually inject there, so created another retinotomy, but he made it too close to the original retinotomy, so most of the drug leaked out of the first retinotomy hole. So one of the things you learn in practice, so we had a criterion that if you are going to make more than one retinotomy to do your injection, make sure it's far away from your first one. In this particular case, we learned why that's what you do. So there's some learning that goes along the way, but out of 42 cases, that's pretty good. And now the issue is scalability. So now we'll be moving in our pivotal development to a lot more surgeons. But we have a surgery training team, and we've learned, there's a lot of pearls we've learned from the existing surgeons in terms of standardizing training and actually performing this. So we actually don't think it's going to be a problem. So it's not a need to go to an office-based, but let's face it, it still is a surgical procedure. So it's the gold standard. It's the best, the most effective, safest way we know to achieve what you want to achieve. But if you could achieve similar safety and efficacy without going to the OR, then that would broaden the patients that you would consider the benefit risk to be appropriate to go ahead with it.
I totally agree with that, and that makes perfect sense. And these are older patients, and they have a natural disinclination to go to the OR themselves. So yes, options are great. It makes sense to me why this would have durability and last. Why wouldn't it continue in perpetuity? What happens that dampens the effect later on, if it does?
Yeah, one consideration outside the retina when people think of, for example, liver-directed gene therapy for things like hemophilia, or if you go for other target tissues where the cells are still dividing, or where there's not end organs with non-dividing cells, is you can have a dilution, or there's at least a theoretical belief that if you could have a dilution of the vector, so the number of gene copies that you have per cell will go down over time if your cells are still dividing, or you have cells dying off and other ones coming. That's one of the benefits of retina, is that these cells are not dividing. So we don't expect there to be any kind of dilutional effect. Even in the liver, we know with hemophilia, now some of the early trials, some of those patients are now 10 years on, and we see good durability. So based on that, and also with Luxturna, with each year that goes by, we have longer and longer follow-up of these patients that supports that we should have very good durability. Ultimately, we're going to have to follow patients in those programs and our own programs to fully characterize that and see, do you have any decrease over time? But at least the available data we have from other clinical trials suggest it should be for a very long time. So certainly for a wet AMD patient, we expect this to be a one and done treatment option. When you start moving into younger patient populations like diabetic retinopathy, for example, that's obviously a longer time horizon for those patients. And we'll have to see, but certainly based on the hemophilia trials, 10 years or longer.
I'm glad you brought up retinopathy. That was what I wanted to ask you about next. Where are you with the diabetic retinopathy program now? Or is that heading into the clinical space and clinical trials now?
Yeah, so in addition to the suprachoroidal delivery for treatment of wet AMD study that we started dosing on earlier this month, we also have announced that we're about to start another study with suprachoroidal delivery for treatment of diabetic retinopathy. So that'll be starting in a matter of weeks. We chose suprachoroidal delivery because we thought, thinking of risk-benefit, particularly if we want to be able to treat asymptomatic patients before they develop the sight-threatening complications, there would be a setting where a office-based approach would be the best to really address the unmet need that exists there right now, where we know Lucentis and Eylea work, and that they not only regress existing diabetic retinopathy severity, but importantly, that translates into a sizable reduction in progression to DME and PDR from the different studies that have been done. But in spite of that, and in spite of the label expansion for both Lucentis and Eylea, we know that most retina specialists are not actually providing this to their NPDR patients unless they have DME, of course. And that's not too surprising if you think of those patients where, yes, you know they're at increased risk of developing the sight-threatening complications, but do you really want to start them on a course of repeated intravitreal injections as opposed to just watching them more closely? So I wonder how you and your group deals with those patients. I imagine there's some, where weighing everything on a patient-by-patient basis, it makes sense, but it seems like in the real world for the majority of patients, given current treatment options, it just isn't making sense.
It's a great question and an unbelievably important point. I actually have a lecture on this, and the data on advanced non-proliferative diabetic retinopathy in the absence of macular edema with Eylea, Lucentis, the Panorama Study, it's phenomenal data. It's really profoundly good data if you view it as a patient yourself with the knowledge of retina and what a clinical trial means and how it works. You would want it for yourself. And every time I give the talk about this, I ask myself and sort of ask the audience at the same time, but why aren't we doing this? And we're not. And it has everything to do with the acceptability of the idea in the clinic to yourself and to the patient. How how realistic is this that I can inject an asymptomatic eye, potentially in perpetuity again, to what endpoint? But it is phenomenal data. And if you can achieve that kind of reversal of diabetic retinopathy with a much less frequent dosing regimen, if not single, but say infrequent, I can't see why we wouldn't do it. The data, the photographs, the clinical reversal of these patients with Eylea or Lucentis given regularly is really incredible. We've never seen this before.
And to have that clinical data that clearly demonstrates that if you block VEGF, you can get these outcomes. So it's just a matter of what's a sustainable way to achieve that.
Yeah.
And if we think of ranibizumab port delivery system, in addition to advancing in other areas, they're advancing in this population in diabetic retinopathy. And that's even with a surgical implant, and albeit simpler refill, still needing to refill, and probably less frequently than every 6 months as you would need for DME and wet AMD, still needing refill. So that's almost so we have the clinical validation. We also have almost a commercial validation of others in this space recognizing this is a big unmet need. So for us, the best way to deal with this would be an in-office one-time treatment. And then you deal with the hurdle that seems to be preventing application of or translation of these great study results into the real world.
It's an amazing conceptualization, and it sounds like a reality, which brings me to maybe the final question. You sort of touched upon this. We both did just now, but I'll give you a chance to elaborate. Where do you see this then going in the near, intermediate, and long term with the coming of other longer acting drugs, or more importantly, drug delivery systems, some of which we're testing now, not just surgical, but also gelatinous materials, pharmaceutical products that can be, used as carrier molecules. With all this coming to extend the duration of these excellent drugs we've developed for retinopathy and for AMD and potentially other things, where do you see the gene therapy fitting in that entire complex of treatments for our patients?
Yeah, it's been great to see the advance of the field in general, with new agents coming along that have the potential to be more durable, to get us from, originally monthly dosing, then Eylea every other month dosing, maybe every 12 weeks. Brolucizumab, of course, before the unfortunate occlusive vasculitis inflammatory aspect, looked to be moving in that direction as well, incrementally getting a little more durability. Some other agents that are interesting, like Kodiak and Graybug, on that continuum of trying to get to every 3, 4, maybe 5 month dosing, and the port delivery system that we discussed of, maybe every 6 month dosing after having the implant in. So I think all these things are going in the right direction. There's still incremental benefits along the way, but I think we're seeing by the fact that these are being developed and that there's great interest in the retinal community to these incremental benefits, supports the concept. So in a way, you can kind of think of them as competition, but they're actually more validation, again, of how valuable this would be for you and your colleagues and your patients to be able to decrease the treatment burden. And really, the ultimate way to do that would be a one-time treatment. I don't think there's never going to be a one-time treatment that treats all the patients, because it's a complex disease, and there are some patients who don't respond to even very frequent anti-VEGF injections. And so I think that's also encouraging for not just how do you keep anti-VEGF around longer, but also some of the other interesting experimental approaches out there, looking at other mechanisms, whether it's, pan-VEGF, like the Opthea program, or faricimab going after another mechanism of action. But I think all those approaches are really going to be incremental benefits in terms of durability. So I think near term, we still got to show it. We got to show it definitively in large studies. So that's kind of the near term step. But for us, if in our pivotal development, we can meet our target product profile of having at least as good a visual acuity and anatomic outcomes while dramatically reducing treatment burden, we believe that's a win and a further big advance over, fortunately, however far we can go with durability with the other agents and the other approaches.
Totally clear, and I agree, and good luck in those studies. And I want to thank you for coming on, Steve. This has been a really very useful review of the entire concept of how to deliver gene therapy to the retina for these great clinical outcomes. REGENXBIO, and you should be congratulated. This is phenomenal stuff. Thank you for sharing it with us today.
Thanks so much, Faris. Thanks for having me.
We're glad you could listen in to our conversation with Steve Pakola, MD. We hope you found it as informative and forward-thinking as we did. For more great retina content, tune in next time. And if you haven't already, subscribe to the OIS Retina podcast at ois.net/subscribe.